We're going to begin Chapter 19 by looking at the basic flow of blood through the heart and the body. You want to follow along with pages 730 and 731 in your book. So go ahead, if you don't have your book out, pause the video, get your book, turn to those pages. There's so many words and so many things to label, it's not practical to write all that out on our picture in your binder. So I'm going to give the ideas and I'm going to use abbreviations. You'll need these pages in your book to get the actual full terms. Another thing to point out is the use of red and blue. So we use blue to represent deoxygenated blood. So it doesn't mean that this blood has zero oxygen in it. Your blood is never at zero oxygen. It has less oxygen because this is blood that has given oxygen to cells. In real life, it is not blue. Your blood is never blue. Deoxygenated blood in real life is dark red. The book just uses blue as a representation. Then we also have parts of this in red. Red is used to represent oxygenated blood. This is blood that has picked up oxygen in the lungs. So it is at a higher level of oxygen. So again, your oxygen's never zero. There's always oxygen in your blood. It can be higher or it can be lower. And your blood is never blue. It's either dark red if it has low oxygen, and oxygenated blood is bright red. So oxygen does increase the redness of the blood, makes it more oxygen in your blood, the brighter red it is. Now let's start looking at the heart. The heart is divided into two sides, and it's always the heart's right and left, not your right and left. So you have to get used to flipping right and left. But we have over here the right side of the heart in blue. So the right side of the heart has deoxygenated blood. And we have over here the left side of the heart in red. So the left side has oxygenated. Then we have the lungs. So here's a lung and here's a lung. The lungs are where you get oxygen. So the blood is going into the lungs as deoxygenated. Here, notice it's purple. This is your pulmonary capillaries. So it's transitioning from deoxygenated to oxygen. It's getting oxygen. Then we also have systemic. This means out in your body. So these are systemic capillaries. You have some at the top, some at the bottom. The ones at the top here represent your head and arms. The things that are above your heart, the ones at the bottom represent the rest of your body, what's below your heart. And here again, it's purple, it's transitioning. This is where you are giving oxygen. So this is where you give oxygen, and lungs is where you get oxygen. So you have kind of this figure 8 pattern. Let's make an analogy here. Let's suppose that you have a job and you get up and you go to work. So you leave your house and you go to your work. So you have your house. And you go to work. And at work, you get money. Then you come back home. Now you're back home with your money. Now you leave your house and you go out to a party. or you go out, not necessarily to a party itself, but you go out with your friends and you spend your money. Maybe you go out to eat or you go to the movies. You do whatever you do to have fun and you spend your money. Then after you spend your money, you come back home. Now you're out of money. So the next day you go to work again and you get more money, you come back home. Then you go out and you spend your money. Now you're out of money, you come back home. You keep doing that day after day. It's this figure 8 pattern. That's the idea with blood flow through the heart. You have deoxygenated blood in your heart. It goes to the lungs and it gets oxygen. Then it comes back to the heart as oxygenated. Now you have oxygenated blood in the heart. This oxygenated blood goes out to systemic, it goes out to your body, and it gives oxygen. Then it comes back to the heart as deoxygenated. So the blood is always going to go from the heart to the lungs, back to the heart, and then from the heart to the body, back to the heart. It's this figure 8 pattern. So we have the right side of the heart receives deoxygenated blood from the body. So you're going to have deoxygenated blood from the body goes into the right heart. The right heart pumps it to the lungs where it will get oxygen. Then you have oxygenated blood to the left heart. Then the left heart pumps it out to the body where it will give oxygen. Then it returns, becomes deoxygenated, and returns here to the right side of the heart. Now we're going to build on that basic pattern. The heart has 4 chambers. Okay, so these are like compartments. You have atria on top, so you have a right and a left atrium on top. So atrium is singular, atria is plural. Then you have your ventricles on the bottom. So you have right and left ventricle on the bottom. Atria receive blood from veins. So veins are what carry blood to your heart, and the atria are the receiving chambers of the heart. Ventricles pump blood into arteries. So arteries are what carry blood away from the heart, and the ventricles are the pumping chambers. They're pushing the blood out into those arteries. So now is when you want to follow along on your book, find the atria and the ventricles. I said it gets too much to try and label all that stuff in our picture here. We can put some little initials here. This would be like your right atrium, right ventricle. Over here is your left atrium. You can't really see that. Left atrium, left ventricle. Blood flows from atria to ventricles. So the path of blood is going to be like this, from the atria to the ventricles. Okay, then let's look at the vessels. You have all these vessels carrying the blood back and forth to the heart. Whenever we're talking about the path of blood through the heart, we're always going to start with the right atrium. That's always our starting point. So we're going to go ahead and we're going to get used to that. And we're going to start with the right atrium. So you're going to have deoxygenated blood in the right atrium. That's going to flow through to the right ventricle. The ventricle is going to pump that blood. And the vessel that it pumps it into is the pulmonary trunk. This is the pulmonary trunk right here. And again, follow along with the pictures in your book to more clearly see the pulmonary trunk. That's where the deoxygenated blood is being pumped from the right ventricle. The pulmonary trunk, if you follow it up, it branches. So you have a branch here and you have a branch here. Those are your pulmonary veins. Pulmonary arteries. Arteries go away from the heart. So this branches into your two pulmonary arteries. The pulmonary arteries are what take that blood into your lungs. And in your lungs, you then flow through your pulmonary capillaries. So here we already labeled your pulmonary capillaries. That's where you get oxygen. Veins carry blood back to the heart. So now we have right here and right here, pulmonary veins. And you have 4 of them, 2 on each side. Those go to your left atrium. Then from the left atrium, we flow into the left ventricle. Then the left ventricle is going to pump into an artery. Right here is the artery the left ventricle pumps into. That is the aorta. The aorta branches, and in chapter 20 we're going to learn the names of a lot of these arteries and veins, but for now we're just going to call them systemic arteries or systemic veins. So you have branches that come off of the aorta. All of these branches that go out to all the parts of the body we can collectively call systemic arteries. So from the aorta, you branch out into systemic arteries. That's what goes out to all the different systemic capillaries all over your body. So all over in your body, you have all these capillary beds. So you just keep branching and branching to get to all the parts of your body. So you're going to go to your systemic capillaries. That's where you're going to give oxygen to your cells. Then we have deoxygenated blood is going to go to your systemic veins. And again, you start off with lots of branches. Hundreds of branches of systemic veins. And they are moving back toward your heart. And as they do, they merge with each other. So you have all these branches, they're merging, forming fewer veins, but those fewer veins are bigger. As you get close to the heart, you come down to just 2 main veins that are going to connect to the heart. You have the superior and inferior vena cava. So all these systemic veins merge in the superior vena cava and inferior vena cava. So the superior, remember top, everything from your head and your arms is coming right here to the superior vena cava. Anything from below your heart is coming here to the inferior vena cava. And those 2 vessels are what put the blood into your right atrium. So there's your path. We can divide this path into 2 circuits. You have a pulmonary circuit and a systemic circuit. The pulmonary circuit is the right side of the heart to the lungs. And the systemic circuit is the left side of the heart to the body. Both circuits begin with an atrium. So pulmonary circuit is going to begin with the right atrium, and it's going to end with the pulmonary veins. The systemic circuit is going to begin with the left atrium, and it is going to end with the superior vena cava and inferior vena cava. Okay, and again, follow along with your book. It gives complete list of everything in these two circuits. Okay, one more thing we need to talk about now is valves. You have to make sure that blood always flows in the right direction. You don't want blood to back up. So you have valves to ensure one-way flow of blood. And these valves are found at the entrance and exit of each ventricle. So you have two AV valves, which stands for atrioventricular. These AV valves, as their name says, are between the atrium ventricles. So these are at the entrance to the ventricle. So this is between the atria and the ventricles. So this little flap here and this flap, and see these little flaps? That's the AV valves. They open to let blood go from the atria into the ventricles. And then when the ventricles pump blood into the arteries, they close. That way the blood has to go into the arteries and not back into the atria. The other set of valves is the semilunar valves. These are between the ventricle and the artery that it pumps into. So you have the pulmonary semilunar, would be right here, where the right ventricle pumps into the pulmonary trunk, and the aortic semilunar is right there, where the left ventricle pumps into the aorta. So if we add in our valves now, and we do a whole entire path of blood through circulation, we're going to start with the right atrium. Then that blood has to flow through the right AV valve. Now, there's other names for these AV valves. The right AV valve has 3 parts to it, so it's also called the tricuspid. Then it would be in the right ventricle. Then the right ventricle has to pump the blood through the pulmonary semilunar valve into the pulmonary trunk. Then that trunk branches into the pulmonary arteries, which goes to the lungs, to the pulmonary capillaries. We'll go ahead and put that in this purple, because now we're getting oxygen. Then from the pulmonary capillaries, we now have oxygenated blood. It's going to go back to the left side of the heart. So now we're going to have pulmonary veins. This whole line so far, all of this is pulmonary circuit. Then the pulmonary veins put that oxygenated blood into the left atrium. It has to flow through the left AV valve. It has 2 flaps, so it's also called the bicuspid, and another name for it is mitral, which you'll see later how it got its name mitral. Then we go into the left ventricle, which pumps that blood through the aortic semilunar valve into the aorta. The aorta branches to go all over your body, so those are systemic arteries, all those branches. Then we go into the systemic capillaries, where you give oxygen to your cells. Then we have deoxygenated blood in your systemic veins. These merge together as they get closer to the heart, and you end up with the superior vena cava and inferior vena cava, putting blood back into your right atrium. And this whole line is the systemic circuit. So practice this. The rest of the chapter, you're going to need to know this flow of blood through the heart and lungs and body. So review this, practice this. You should be able to list it all out. If I say start at the right atrium, you should be able to trace the path of blood and follow it on a picture all the way through the heart, to the lungs, back to the heart, through the body, and back to the heart again. In this section, we're going to learn about the 3 layers of the heart wall and the pericardium. So let's begin from the inside and work our way out. Inside, the heart has a thin coating of endocardium. It lines the whole inside. And we're looking at cross-section here, so I'm just doing it in the cross-section. So here it is in the ventricles. Here it is up in the right atrium. And you'll see this in lab when we dissect the heart. But all of that is endocardium. It is just this thin coating. Think of it like the paint on a wall. It's just this thin coating. And it is very smooth. That's the whole point of endocardium, is it's smooth. And this allows blood to easily flow over it. And it is continuous with the lining of the blood vessels. So everywhere your blood's flowing, inside your heart, inside your blood vessels, is this smooth coating. Then next we have myocardium. That's the majority of the heart wall. Think of, like I said, if endocardium is paint, myocardium is the wall itself. Myocardium is the muscle of the heart. And one thing to notice about it, if you compare the right and the left, remember this is right, this is left. It's the heart's right and left, not your right and left. It's much thicker in the left. Notice the thickness of the myocardium here versus here. So it is thickest and therefore strongest, right? Think about what you know about muscles. Strong muscles are bigger in the left ventricle. This is going to help you out in lab when you dissect the heart, because the real heart, when you're dissecting it, it's not going to look perfect like this picture. And the first thing you need to do is figure out right from left. And the way you figure that out is by comparing the thickness of the myocardium. So the place where you find the thick myocardium, you'll know is the left ventricle. You think about the reason for this. The right ventricle, where does it pump blood to? Remember, the right ventricle pumps up through, here's our pulmonary semilunar valve, into the pulmonary trunk, which branches to the pulmonary arteries and goes to the lungs. The lungs are next to the heart. That's it. That's as far as that blood has to get, is the lungs. The left ventricle is going to pump through there. That's the aortic semilunar valve, up to here to the aorta, and then it branches out to the entire body. So all the way to the top of your head, the tips of your fingers, the tips of your toes. That's a lot of work. This is a much harder job. So the left ventricle is stronger so that it can pump blood to the entire body. Then we'll move on to our next layer, is out here on the surface of the heart. And this is epicardium. So it's just like endocardium. Imagine you have a wall that is painted on both sides. And just like endocardium, it is also smooth and shiny. And you'll see this in lab when you dissect the heart. You can see that the inside surface and the outside surface are kind of shiny. That's the endocardium and epicardium. And this is smooth because your heart is beating, it's moving, and this is smooth in order to make it easy so you don't get a lot of friction when the heart beats. So that's the 3 layers of the heart. Now let's talk about the pericardium. Pericardium is the tissue that surrounds the heart. We can divide this up into parts to think about. So we're going to have the visceral layer of serous pericardium. Then we're going to have the pericardial sac. And this is divided into 2 parts. We have the parietal layer of serous pericardium. And then you also have the pericardial, or the fibrous pericardium. Between the visceral serous pericardium and the parietal layer of serous pericardium is the pericardial cavity. Okay, so let me get my words all straight this time. Let's come over here and label these. So notice for the visceral serous pericardium, I used the same color as epicardium. That is because these 2 things are the same. It's 2 different names for the same thing. If you are talking about layers of the heart wall, you call it epicardium. If you are talking about the pericardium, you call it the visceral layer of the serous pericardium. Kind of think about when I'm at school, I'm a teacher. When I'm at home, I'm mom. I'm the same person, but I have 2 different titles depending on where I'm at, what context. So this is the same exact structure, it just has 2 different titles depending on which context you're talking about. Then there is the parietal layer of serous pericardium. That is this part. The 2 layers of serous are one sheet of tissue that is folded in half. If you look up here at the top very closely, we can see here's our visceral, here's our parietal, and see where they meet? It's actually one thing folded in half. Between them is that pericardial cavity. Here's the cavity. It's the space between them. And notice the heart's not in the pericardial cavity. The heart is in the mediastinum. Then the fibrous layer is out here. This is the fibrous layer. And it comes down, it attaches to the diaphragm and it attaches to the arteries. So it's over here, you can see it starts off, it comes down and it attaches to the diaphragm. So one thing to notice is that the heart is not in the pericardial cavity. A lot of people, your first instinct is to think that it is, but it is not. And if you remember in chapter 1, we kind of touched on this in chapter 1, that breakout activity you did, this was one of the questions on there. So if you have a really good memory, you might remember that. The pericardial cavity surrounds the heart, and it is filled with serous fluid. So the serous pericardium, it's a serous membrane. You have 3 serous membranes in your body. If you go back to chapter 1, all the way back to page 15, it shows you this. You have 3 serous membranes. The pericardium around the heart, the pleura around the lungs, and the peritoneum around your abdominal organs. A serous membrane is a sheet of tissue folded in half so that you get 2 layers, and you have a cavity between the 2 layers, and the membrane secretes a fluid that fills that cavity. And the purpose of the fluid is to lubricate. So again, remember, the heart is beating, it's moving. You don't want a lot of friction, so you lubricate that area. And the correct answer of which cavity the heart is in is mediastinum. So this is a specific area within the thoracic cavity that contains the heart. So I have another little video clip that's going to be spliced in here where I have a plastic heart and a sheet of felt paper, felt cloth, and a paper towel, and I kind of show you this a little better. Hello, I'm going to show you why the heart is not in the pericardial cavity. Let's suppose this is the serous membrane. Serous membrane is a double layer, so we have to take this flat sheet of tissue and fold it in half. Now we have our 2 layers. This then gets wrapped around the heart like this. The inner layer that is touching the heart is the visceral layer of the serous pericardium. This outer layer that's not touching the heart is the parietal layer. Between the 2 layers is the pericardial cavity. Notice that the cavity is surrounding the heart. The cavity is this area here between the 2 layers. That cavity wraps around the heart. The heart is not in the cavity. If the heart were in the cavity, it would be like this, but it's not. The heart's here. The only thing in the cavity is serous fluid. Then you have the fibrous layer, so we'll pretend this paper towel is a fibrous layer. It goes on top like this. So your pericardium, you have the fibrous layer, you have your serous, which is folded in half so that you have a parietal layer out here, a visceral layer that's touching the surface of the heart, and the pericardial cavity between those 2 layers of the serous membrane. The fibrous skeleton and gap junctions are a couple important features of the heart that help it to function correctly. First, let's see what a fibrous skeleton is. This is connective tissue in the heart. Your skeletal muscles are attached to your bones. Your heart doesn't have any bones in it for the muscle tissue of the heart to attach to. So instead it has this connective tissue framework that the muscle cells attach to. So it provides the framework. It also has a couple of other important jobs. It provides electric insulation, between the atria and the ventricles, and it controls or it contributes to the way the ventricles contract. It causes a slight twist in the ventricles as they contract. So we'll look at these in some more detail. When the heart beats, it's a two-part beat. First, the atria contract. So in this picture, you can see these lines here on the side. They represent that it's contracting, and you can see the atria is kind of dented in. That represents the atria contracting. Notice while they're contracting, the ventricle is relaxed. Then step 2, the ventricles contract. So here you can see the ventricles are contracting. And while the ventricles are contracting, the atria are relaxed. So this taking turns is very important. Atria and ventricles take turns contracting. You never want them both to contract at the same time. And when I say both, I mean atria and ventricles. The right and left stay coordinated with each other. So step 1 of a heartbeat is that both the right and left atria contract. And during this time, the ventricles are relaxed. Then step 2, both the right and left ventricles contract. And during this time, the atria are relaxed. The fibrous skeleton provides insulation between atria and ventricles. It's an electric signal that stimulates the contraction. And the fibrous skeleton insulates between the atria and ventricles so that that signal can't spread from atria to ventricles and vice versa. Now, with this electric signal, though, you do want it to spread between right and left sides. Notice step 1, the right and left atria both contract. You want them to be synchronized with each other. The same when it comes to the two ventricles. The right and left ventricles both contract at the same time. So our other feature here is gap junctions. This is a picture of cardiac muscle cells, and you have cells connected to their neighbors. Like right here, this squiggly line, that's where cells connect to each other. So they connect end to end. And where they connect, the structure that connects them is called an intercalated disc. So cardiac muscle cells attached to their neighbors by intercalated disc. Think of like the button on your pants. That's what holds the two sides of your pants together. So intercalated discs are kind of like a button. They hold the two cells together. In the center of each intercalated disc is a hole. So these are your intercalated disc, and there's a hole in the middle. That is the gap junction. Electric signals, if you remember from chapter 12, are ions. It's sodium and potassium. And in the heart, it's also calcium. So when we're talking about electric signal in the heart, we're talking about sodium, potassium, and calcium flowing, moving in and out. These holes allow those ions to move between cells. So if one cell has an electric signal, so does its neighbor. So all the cells that are connected to each other will experience an electric signal together. So it's because of these gap junctions that the right and left atria contract together. They're in synchrony with each other. You know, the Olympics, when they have the synchronized swimmers, that's how your heart muscle cells are. And the right and left ventricles contract together. So you have two things going on here. Gap junctions allow the right and left atria to share the signal with each other and contract together, while the fibrous skeleton blocks that signal from spreading down into the ventricles. That way the ventricles stay relaxed while the atria contract. Then we just flip it. The ventricles contract and they use their gap junctions to share the signal with each other, so both the right and left contract together, while the fibrous skeleton blocks the signal from going up into the atria. That way the atria stay relaxed while the ventricles contract. Okay, then back to the other job of the fibrous skeleton. The structure of the fibrous skeleton is kind of a spiral. It's not like your walls where it's 90-degree angles. It has this twisting spiral arrangement. So when the heart contracts and those cells shorten, they follow this spiral arrangement. So the ventricles, especially, the ventricles are the ones that are pumping blood up into the arteries. As the ventricles contract, it's a slight twist to it. You can think of it like wringing out a towel. If you take a towel, a wet towel, and you squeeze it, you'll get some water out. But if you squeeze and twist, you'll get even more water out. So this spiral arrangement causes the ventricles to slightly twist as they contract. This means that they're more efficient at pushing that blood into the arteries. Just like twisting a wet towel makes you more efficient at pushing the water out of it. So this allows them to push more blood into the arteries. Now we're going to look at the energy needs of the heart. The heart has a very high metabolism, so it uses lots of ATP. Right? It has to work 24/7, your whole entire life. It never gets to take a break or rest. So it's going through massive amounts of ATP, keeping you alive. Now think back to chapter 3 and what you learned about ATP. In order to make ATP, the cell needs certain things. you have to have a fuel source. Now, back in chapter 3, we talked about glucose and fat as fuel sources. You also need oxygen. The more oxygen you have, then the more you can keep going in cellular respiration and not have to switch over to fermentation. And you get more ATP when you have oxygen. The third thing you need is mitochondria. Mitochondria are the organelles where cellular respiration occurs and where the majority of ATP comes from. Remember, glycolysis occurs outside the mitochondria and gives you 2 ATP, but then once you go into the mitochondria, you get another 34 ATP. So the heart has features to maximize these. If you maximize these 3 things, then you will maximize your ATP production. So let's look at the features of the heart. The first one here, this menu, says, I'm so hungry I'll take one of everything on the menu. And if you look at the menu for the heart, you have glucose, fat, lactic acid, ketones, amino acids. This goes beyond just the glucose and fat that we learned about in chapter 3. The heart is not picky. It will use a variety of fuel molecules. So number 1 here is variety of fuels. It's kind of the opposite of neurons. Neurons will only use glucose. They're very picky. The heart is not picky at all. The heart is like the one who's hungry and will eat anything. Even some of these things are waste products from other cells, like the lactic acid and the ketones. That's waste that other cells put into the blood, and the heart's like, hey, you're not going to eat that? I'll take it. And it uses those. When it comes to oxygen, you have 2 things that help you out with oxygen. So number 2 and number 3 here both go along with oxygen. You have extensive coronary circulation. Yes, the heart is filled with blood, but the blood that's in it is there to be pumped. It's not being nourished by the blood. Right here is the aorta. Remember, the oxygenated blood from the left ventricle is being pumped into the aorta. And you can see right here at the base of the aorta, on each side, are your coronary arteries. They are the first branches to come off from the aorta there with oxygenated blood. And they branch out onto the surface of the heart and deliver oxygenated blood to all of the heart muscle cells. Your heart also has lots of myoglobin. Hopefully you remember this from chapter 10 about muscles. We first learned about myoglobin in muscles. Myoglobin is similar to hemoglobin. Hemoglobin's in the blood, myoglobin's in the muscle. Myoglobin stores oxygen. And it's red. So that's what gives meat its red color. The more red a meat is, the more myoglobin it is. Your heart is very much red. And the reason that it's red is because it has myoglobin. So when you are resting and you're not using a lot of oxygen, your heart hoards it. It's like an episode of Hoarders. Your heart has all this myoglobin and it's grabbing all this oxygen and hoarding it. Then when you're active and your heart needs that oxygen, it has it to use. And getting oxygen to your heart is very, very important. Ischemia is a condition in which you would not get enough oxygen. And remember, other cells would switch to fermentation. Well, your heart can't survive on fermentation. That's not enough ATP for it. So if you get ischemia, not enough oxygen to your heart cells, that is what can lead to a heart attack. So getting plenty of oxygen to your heart is very important. And then with mitochondria. So right here it says, look at all my mitochondria. Here's a mitochondria. Heart cardiac muscle cells have the most mitochondria of any cell in the body. In fact, they are 25% mitochondria. That is more than any other cell in the body. So your heart is packed full of mitochondria to make lots of ATP to keep it going. You know about coronary circulation. You have these arteries, and there are also veins on the surface of the heart that are there to supply oxygenated blood to the cardiac muscle. You also know how important it is to get plenty of oxygen to the heart because the heart cannot tolerate ischemia. So you have this system of these coronary arteries that branch off here from the base of the aorta to take oxygenated blood to the cardiac muscle. This is a great system and it usually works just fine. But there is what you could kind of think of as a design flaw in here. You also know about the valves. You have valves controlling the flow of blood. Where the aorta attaches to the left ventricle, you have the aortic semilunar valve. So this is in the base of the aorta. Every time the ventricle pumps, this valve opens. So this picture shows it open. These flaps here are the flaps of the valve. And you can see it's open and blood is flowing through into the aorta. This is what happens when the ventricle contracts. And contraction is systole. So we'll go ahead and put ventricular systole. So the aortic semilunar valve is open during ventricular systole. In the next picture, you can see it's closed. So here's the valve. It's these white flaps, and it's closed. That way blood doesn't flow from the aorta back into the ventricle. So it is closed during ventricular diastole. Diastole means relaxed. So when the ventricle is relaxed, it's not contracted, that's when this valve is closed. Now look at the direction the valve opens. It opens upward. Remember, these coronary arteries are coming out of the base of the aorta. So when it opens, right here are where those coronary arteries come out. The flaps of the valve block those coronary arteries. And when it's closed, those coronary arteries would be here like this. Blood is free to flow into the coronary arteries. So when it's open, it blocks the coronary arteries. And when it's closed, blood flows into the coronary arteries. So what this means is that every time your ventricle contracts, that aortic semilunar valve opens. And when it opens, it blocks the coronary arteries. And if those arteries are blocked, you are not sending oxygenated blood to your cardiac muscle. So you have an interruption in the flow of oxygenated blood to your cardiac muscle. So you can actually have a heart attack if your heart beats too fast. Every time your heart beats, you block this. So let's say the average adult resting heart rate, 75 beats per minute. This means that in 60 seconds and 1 minute, you interrupt the blood flow 75 times. Now, you can live with this. You have the myoglobin, your body's adapted, you can handle this. But there is a maximum. You may have heard of the 220 rule. It's just a general rule of thumb, right? So it's not an absolute number. It's not like if you go over this number, you're going to instantly fall over dead. But in general, just to give you a rough idea, your maximum heart rate is 220 minus your age. So if you are 20 years old, you could safely get your heart rate up to 200 beats per minute. Right? And the older you get, the less of the heart rate you can handle. There's an interesting phenomenon that goes along with this called the mammalian diving reflex. And this is found in all mammals. And remember from chapter 13, learning cranial nerves, you have the trigeminal nerve. This nerve is in your face, and it senses temperature of your face. It responds to cold water touching your face. So if cold water touches your face, your trigeminal nerve will send a signal to the medulla oblongata. And your medulla oblongata will then send a signal through the vagus nerve, hopefully you remember this one from chapter 13 also, to secrete acetylcholine onto your heart, and this will cause your heart to slow down. So there's this show, Man vs. Wild, with Bear Grylls. He's a survival expert, and he goes around teaching you how to survive in different situations. He did an episode where he discusses this mammalian diving reflex. So we'll watch what goes on with Bear here.
Okay. Heart rate, but also my breathing rate.
It's all linked up to a laptop, so the team can monitor me in real time.
And I've got a thermal imaging camera showing my skin temperature. Okay, let's get into this.
My skin temperature's currently 88 degrees Fahrenheit. That's about to fall dramatically. I've got to swim over 80 feet. Half of that is under solid ice.
Okay, let's go for this. This is much harder than normal. Whenever I've done this before, it's been part of a journey. You come across a river, you just get on and do it. This is much more premeditated. I find that harder. Okay, first you've got to do is fight that sense of panic that's very natural when you're in freezing cold water like this. And instantly I feel my heart rate just goes straight through the roof. And the danger of that is cardiac arrest.
So let's talk for a minute about what he just said. He said, fight that sense of panic. So as soon as you jump into cold water, that gives you a fight or flight. He is having a sympathetic response here. His blood is full of norepinephrine. He is dumping massive amounts of norepinephrine onto his heart, and his heart rate is increasing. And he just said the danger of that is cardiac arrest. Well, cardiac arrest is another way of saying heart attack. So he's saying he's panicking, which is making his heart rate go up. And that increased heart rate means that every time that ventricle contracts, he's blocking the blood flow into his coronary arteries, and he could actually exceed that maximum safe heart rate and not get enough oxygenated blood to his heart and have cardiac arrest as a result of his heart beating too fast.
You gotta try and slow it. Slow it and keep that panic away. Okay, under the ice. The danger of me here, guys, is not getting the line right. You know, if I start heading offline, you gotta keep eyes on me, okay? We got no safety liner here. You need to get it straight.
Survival is all about understanding and controlling your reflexes. Watch as my heart rate drops from 160 beats per minute to 55. It's known as a mammalian dive reflex. When your face is submerged in cold water, the heart slows dramatically. It gives you vital extra seconds to make it to the surface.
All right, so as soon as he put his face in the water, his heart rate dropped from 160 to 55. That is a very rapid and huge decrease, and that is the mammalian diving reflex. Every mammal will do this. You put cold water on your face, it slows your heart rate down. You've learned some of the features of the muscle of the heart. Now let's look at the nervous tissue in the heart. The heart is autorhythmic. This means it stimulates itself to beat. It has its own built-in system that makes it beat. It does not rely on the brain. Your brain does not tell your heart to beat. Your brain can tell your heart to beat faster or slower, but it doesn't tell it to beat. That happens internally. And the structure inside that does that is the cardiac conduction system. In fact, you can take the heart out of the body and it will still beat. I have a little video here to show you that.
Put it in a solution. We wanted one color.
And if you ever go hunting or fishing, if you clean an animal, you might notice that you can take the heart out and it will still keep beating for quite a while in some cases. That's because the cardiac conduction system is doing that. So let's look at the parts of the cardiac conduction system. We start right here. This dot in the top of the right atrium is number 1. This is the sinoatrial node. And it's called the SA node for short. So this is in the top of the right atrium. This is what makes the signal. And down here, we'll look more at how it works once we've gone through all of the parts. Then it sends the signal to part 2. So this dot is part number 2, and the signal travels. The second part is the atrioventricular node. And it is called the AV node for short. This is in the floor of the right atrium. So the signal moves from the SA node to the AV node. As it does, both atria contract. Remember those gap junctions that connect all the muscle cells and allow them to share ions? The signal spreads through those gap junctions. So both your right and left atria contract during this time. And remember, contract is systole. So as the signal moves from the SA to the AV, the atria is systole. And remember also we talked about the importance of the atria and the ventricles taking turns. So next you're going to have the AV delay. Once the AV node receives a signal, it kind of slows it down. It holds on to it for just a fraction of a second before it sends it on down to the ventricles. So this gives the atria time to finish their systole before the ventricles start, because that taking turns is so important. Then we go down, part 3, it's right here. This is the bundle branches or bundle of His. So next the signal moves down this direction. And it's going this way toward the apex of the heart. The apex is that pointy to the tip of the heart. This is the wall that is separating the two ventricles, so this is the interventricular septum. So it goes down the interventricular septum to the apex. Then once it gets to the apex, we have these parts, so here, and then they're over on this side also. Those are the Purkinje fibers. And once you get there, the signal starts going back up. So the signal goes down like this, as it's going down the bundle branches through that interventricular septum. When it gets to the apex, then it goes up the sides like this. So the signal moves up the sides of the ventricles. And now is when it spreads through the gap junctions of the ventricles. And when your ventricles contract, we have an early systole and late systole. So the ventricular contraction begins here at the apex. So first, this part of the ventricle will contract. Then the upper part of the ventricle contract. So we'll say contract early, contract late. So the contraction, it's like squeezing a tube of toothpaste. How you start at the bottom and you work your way up. That's how the ventricles contract. Remember, up here are your arteries. Like here is your aorta. And here's your pulmonary trunk. The ventricles are pushing blood up. So they start at the bottom. So the ventricular contraction begins at the apex and moves up. Okay, so these are the parts of the conduction system. You have the SA node is known as your pacemaker. It's what sets your heart rate. It creates the signal, sends it to the AV node. As the signal moves from SA to AV, it spreads through gap junctions and both atria contract. You have the AV delay, where you give the atria time to finish. Then you send it down the bundle of His, which goes down the interventricular septum toward the apex. You get to the apex, you do a U-turn, you go up the sides of the ventricles and the Purkinje fibers. That's when it spreads through the gap junctions in the ventricles. The ventricles contract, but they begin their contraction at the apex and the contraction moves up, like squeezing a tube of toothpaste from the bottom up. Now we can look at a nodal cell. So this picture is our nodal cell. So this is showing what's going on inside that SA node to create the signal. And you have to think back to chapter 12. Remember when you learned about neurons and how you have a resting membrane potential, and then sodium comes in and it raises the voltage, and potassium goes out and lowers the voltage? We have the same thing here. We're just also adding in calcium. So you can see here on your nodal cell, you have all these gates, right? You can see them all along here. You have gates, just like you had in your neuron. So we have, here is, the pink is sodium. And you can see sodium comes in. So you're going to have sodium in. And here is potassium going out. This part is the same as neurons. It's what you learned in chapter 12. Sodium in, potassium out. We also have now calcium. Calcium comes in. So let's look at the cycle and the voltages. Remember resting membrane potential, the voltage you start at? We're going to start at -60. And that -60, remember voltage regulated gates, they open based on what the voltage is. If the voltage is -60, that makes the sodium gates open. So when we're at -60 here, then you will have sodium come in. Let's make a little number scale here and keep track of where we are. We're down here at -60. When sodium comes in, that raises voltage. Sodium is positive. So sodium is going to raise voltage. Will we raise it to -40? So now we are at -40. That causes the calcium gate to open. Calcium is set to open once voltage hits -40. and calcium comes in. Well, we have more positive ion coming in. So we're going to, again, raise our voltage. This time we raise it up to 0. Zero stimulates the potassium gate to open. And potassium goes out. And when potassium goes out, you reduce your voltage. And we reduce it back down to -60, which then repeats the cycle. Now that makes the sodium open and you start over again. So this is why the heart is autorhythmic. It's this self-perpetuating cycle. It keeps itself going. The last step, letting potassium out, going to -60, is what causes the first step, letting sodium in. Now that you know about the cardiac conduction system and how it stimulates the heart to beat, let's look at regulating the heartbeat. So remember, the heart is autorhythmic. It stimulates itself to beat, but the brain can regulate it. It can tell it to go faster or slower. And if you think back to chapter 13, when we learned about the parts of the brain, the medulla oblongata is in charge of this. So it's going to regulate your heartbeat. It has two different parts here that regulate the heartbeat. You have the cardioacceleratory center, and if you remember to chapter 15, it's the autonomic nervous system, sympathetic and parasympathetic, that are going to do this. So the cardioacceleratory center is going to give sympathetic stimulation to your heart. Then you also have the cardioinhibitory center. This gives parasympathetic stimulation to your heart. So let's look at these. If we do the sympathetic, remember this structure here from chapter 14? That is the sympathetic trunk. So anytime you see the sympathetic trunk, you know you're dealing with sympathetic stimulation. So this is our sympathetic nerve. And if we follow this sympathetic nerve, we get down here to the heart. Let's follow where it branches. It has a branch here to the SA node. Has a branch here to the AV node, and it has branches that come down here to the ventricles. So for sympathetic, you're going to secrete norepinephrine onto the nodes. And this will increase your heart rate. When you put norepinephrine on the nodes, they're going to signal faster and your heart is going to beat faster. It's also putting norepinephrine onto your ventricles. And notice it's not there where the Purkinje fibers are. It's out on the muscle. This makes the muscle contract harder. So think back to chapter 15. Sympathetic causes the heart to beat faster and with more force. So the norepinephrine on the ventricles will make the muscle contract with more force. That's why when you're having an adrenaline rush, you can feel your heart pounding in your chest because you're getting more force out of those ventricles. If we look at the sympathetic is coming down right here. This is the vagus nerve. So for parasympathetic, we're going to use the vagus. So remember in chapter 13 when you learned all the cranial nerves and you learned the vagus nerve? So the vagus nerve secretes acetylcholine onto the node. And when you put acetylcholine on the node, that slows the heart rate. Think back to chapter 15. Look at your chapter 15 lab if you have it handy. For parasympathetic stimulation of the heart, it's decreases rate only. Notice that the vagus nerve does not go down to the ventricles, so it has no effect on force. So sympathetic increases rate and force, parasympathetic decreases rate. So your medulla oblongata is regulating your heart. And it's constantly deciding if it wants to use its cardioacceleratory center and use sympathetic to secrete norepinephrine and increase the rate and force, or if it wants to use the cardioinhibitory center to have the vagus nerve secrete acetylcholine and decrease the rate. So in order to make this decision, your medulla oblongata needs information. So let's think about what kind of information it would base this decision on. Think back now to chapter 1, when we did that chapter 1 lab. So go way back in your mind to the very first lab you ever did in anatomy and physiology. You had 2 beakers of bromethyl blue, and you had a straw, and you blew bubbles into the first one until it turned yellow. Then you went and ran around up and down the stairs or up and down the hall. You got some exercise. And then you blew bubbles again into the second one and timed how long it turned until it turned yellow. The point of that was to compare how much carbon dioxide you're exhaling when you're resting or you're exercising. Now think about why carbon dioxide is so important. Think back to chapter 3 when we learned about cellular respiration. Every time your cells make ATP, they put carbon dioxide into your blood. What happens when the CO2 gets into your blood? Go back to your chapter 3 notes. It mixes with the water in your blood and forms carbonic acid. Now think back to chapter 2 when you learned about pH and acids and bases. If you form acid, this will reduce your pH. Now also think back to early last semester. You have to keep your blood pH at 7.4. This is what your medulla oblongata is concerned with, keeping your blood pH at 7.4. So it has receptors. You have up here in the medulla oblongata is your central chemoreceptor. This monitors the pH of your cerebral spinal fluid. So remember, cerebral spinal fluid, that's the fluid that's made from the blood, and it flows through the brain and the spine. And the pH of this is very important also. You also want to keep it at 7.4. And the pH of this also depends on carbon dioxide level. So your medulla oblongata has a central chemoreceptor that's monitoring the pH of your cerebral spinal fluid. Then you also have peripheral chemoreceptors. Here in your carotids, and here in your aorta. So here you have the peripheral chemoreceptors. They monitor the pH of your blood, the carbon dioxide level of your blood, and they respond to very low oxygen of your blood. Now, it's the pH and the carbon dioxide that we're most concerned with. It's only if your oxygen drops very low that they're going to respond to that. In a healthy person in normal circumstances, your oxygen is not going to drop low enough to stimulate these receptors. So we're not going to worry about those for now. You also have in your peripheral, the carotids and the aorta, you have baroreceptors. These detect how much your arteries are stretching. And the higher your blood pressure, the more your arteries stretch. So now we can look at all the information the medulla oblongata is receiving. You have central chemoreceptor telling it the pH of cerebral spinal fluid. You have peripheral chemoreceptors telling it the pH of blood and how much carbon dioxide is in the blood. And you have peripheral baroreceptors telling it what your blood pressure is. So now the medulla oblongata has to use this information and decide if it wants to send sympathetic or parasympathetic stimulation to the heart. So let's walk through the logic here of how it makes this decision. If we use sympathetic, sympathetic is going to increase your heart rate and your force. This means you will send more blood through your lungs. If you're sending more blood through your lungs, you are going to remove more carbon dioxide. So think about what kind of situation would cause you to want to remove more carbon dioxide. The stimulus and the result have to match up to each other. Your result is that you remove more carbon dioxide. Well, what does that do to pH? Remember, carbon dioxide forms acid in your blood, which lowers pH. If you take away the carbon dioxide, it's not there to form the acid, which means that you increase the pH. Well, if you're wanting to increase your pH, that means there's something going on in your body that's lowering it. Remember, you always want to stay at homeostasis. Your goal is to stay at 7.4. So you would do this if your pH is too low. So if your pH is too low, your medulla oblongata would use sympathetic to increase your heart rate and force. You'll send more blood through your lungs, you'll get rid of carbon dioxide, and you'll bring your pH back up. And this way, you'll stay at 7.4. So think about it. This is when you're active. If you are active, your cells are making more ATP. And if they're making more ATP, they're putting more carbon dioxide into your blood. And if they're putting more carbon dioxide into your blood, that's making more acid and lowering your pH. So those chemoreceptors detect this. They tell your medulla oblongata, it says, hey, I better use the cardioacceleratory center, have sympathetic, send some norepinephrine down there, get the heart going a little more, get rid of this extra carbon dioxide, bring the pH back to 7.4. This would also be with the baroreceptors. Remember, they detect your blood pressure. If they detect that blood pressure is too low, because if you increase your heart rate and increase the force, that will increase your blood pressure and counteract the pressure being too low. Then let's look at parasympathetic. Remember, parasympathetic is going to decrease your heart rate. If you decrease your heart rate, you will send less blood through your lungs. If you send less blood through your lungs, you are going to retain carbon dioxide. And if you retain carbon dioxide, there's going to be more of it there, making acid, which is going to lower your pH. So remember, the results have to match the stimulus. What would cause your medulla oblongata to decide to do this? Well, it would decide to do this if your pH is going up too high. So when you are resting, your cells only make a little bit of ATP. Which means that they're not putting very much carbon dioxide into your blood. So you have low CO2 entering their blood. So there's not very much there to make the acid. So therefore you want to take less out. If less is going in, you want less coming out. You always want to make sure your overall goal is to always have the CO2 in equal to the CO2 out. If whatever goes in equals what comes out, then you will stay at 7.4. So that's why when you're resting, you slow your heart rate. This would also be if the baroreceptors are detecting that blood pressure is too high. Then they could tell your medulla oblongata that your arteries are being stretched too much. Your medulla oblongata will have the vagus nerve secrete acetylcholine onto the node, slow the heart rate down, and if you slow the heart down, blood pressure comes down. So take some time to really review this information. Go back and review the old chapters that this relates to. Make sure this makes sense. You might even want to re-watch this part. Make sure that this makes sense before you go on to the next parts. Now that you know the basics of how the autonomic nervous system affects the heart, let's look at a little more detail and exactly how it brings about those effects. So the sympathetic, remember, is going to act on both the heart rate the force of contraction because it branches down to the node and the ventricles. The parasympathetic is going to act on heart rate only because it goes just to the nodes. So down here we have this for parasympathetic because notice here is a picture of a nodal cell. Parasympathetic is node only, and parasympathetic is going to slow it down. It decreases heart rate. This next section is for sympathetic because it has a node and it has a picture of muscle. You should recognize this picture from chapter 10. That's what it looks like inside a muscle cell. That's the sarcomere. So just like the sarcomeres you learned about for skeletal muscle in chapter 10, you have sarcomeres in cardiac muscle also. And remember, sympathetic is going to increase heart rate by acting on the node, and it's going to increase force by acting on the muscle. And when you increase force, you increase stroke volume. SV is stroke volume. This is the amount of blood that the ventricle pumps into the artery per beat. And it's not total for the whole heart, it's per ventricle. So each ventricle has its own stroke volume. So you have the stroke volume of the right ventricle pumping into the pulmonary trunk and the stroke volume of the left ventricle pumping into the aorta. And the way you alter stroke volume is by altering force. So we're going to talk about how sympathetic alters the force, and then that ultimately is going to alter the stroke volume. Another thing to keep in mind is to keep these things separate. They are two separate things. Heart rate is the number of beats per minute, and stroke volume is the amount of blood pumped per beat. Students tend to want to use these interchangeably or mix them up, like start off a sentence starting about stroke volume and then finish the sentence with heart rate. Don't do that. Keep them separate from each other. So now let's look at exactly how these items are going to work. So remember, we have that cycle. We have -60. That causes the sodium gates to open, and sodium comes in, which brings you up to -40. This causes the calcium gates to open, and calcium comes in, which brings you to 0, which causes the potassium gates to open. Potassium goes out, and you return to -60. If we look at that on a number scale, you have your -60, your -40, your 0. So the sodium brings you from -60 up to -40. The calcium brings you from -40 up to 0. And then potassium takes you back down to -60, and then you start over again. Acetylcholine. So remember, parasympathetic is going to secrete acetylcholine onto here from the vagus nerve. It's going to slow this down. It's going to make this take longer. So if you think about this, what could make it take longer? Well, if we make this a little more negative, let's say we go down to -70, just to kind of pick a number that's less than -60. If you do that, then the sodium has more work to do. It's going to take longer for sodium to get up to -40 if you're starting at negative 70 than if you started at negative 60. You essentially move back the starting line. So if you can move back the starting line, then it's going to take longer for sodium to do its job. So think about how you would move back the starting line. Well, potassium is what lowers the voltage. If potassium goes out and you go down to negative 60, well, let's say now we want to get to negative 70. You would accomplish that by letting more potassium out. You would have to keep those potassium gates open. So if you let more potassium out, you will hyperpolarize. Okay, think back to chapter 12. You learned about hyperpolarize. This lower number is the hyperpolarize. So if you can open extra potassium gates, let extra potassium out, that will make it more negative, hyperpolarize, and then it will take longer for sodium to bring you up to negative 40. That is what acetylcholine does. Acetylcholine acts here. Acetylcholine opens potassium gates. And when that happens, you have more potassium out. And so instead of going to negative 60, you hyperpolarize. Now, it's not always necessarily exactly negative 70. I'm just using that number as an example to give you the idea that you hyperpolarize. You make it more negative. So then you've moved back the starting line for sodium. Sodium has to start at a lower number. So this part takes longer. And you have therefore slowed the heart rate. Okay, so that is the effect of parasympathetic. Parasympathetic reduces heart rate. Remember that parasympathetic does not go to the muscle. Remember down here, the vagus nerve does not branch down to the ventricles. So parasympathetic has no effect on force. Now let's look at sympathetic. Sympathetic is going to end up increasing heart rate, making the node go faster. So again, we have that cycle. We have negative 60, which causes the sodium gates to open. Sodium comes in, and you go to negative 40. This causes the calcium gates to open. Calcium comes in, you go to 0. This causes the potassium gates to open. Potassium goes out, you return to negative 60. Here it is on the number scale again. Sodium brings you from negative 60 to negative 40. Calcium brings you from negative 40 to 0. Potassium takes you back down to negative 60. Now we want to make this go faster. We're going to make this go faster by opening more calcium gates. So norepinephrine is going to act here. Norepinephrine opens extra calcium gates. So you have more calcium in. So that means this part from negative 40 to 0 happens faster. So we're going to have more calcium coming in. So we're going to get from negative 40 to 0 faster. And that's how you increase the heart rate. If we look at the muscle, you're going to have to think back to chapter 10 and what you learned about how muscle contracts. Remember you have the actin and the myosin, and the myosins bind to the actin and pull them in, and then the Z disc is out here attached to that, and that pulls the Z disc in, and the sarcomere is shorter and the muscle is contracted. If you don't remember that, go back and review chapter 10. You have to understand that sliding filament theory. Remember also tropomyosin and troponin. Tropomyosin is blocking the myosin and actin from binding to each other. You have to move it. The way you move it is by having calcium bind troponin. When calcium binds troponin, it moves tropomyosin so that you can then form a cross bridge between myosin and actin. So remember, calcium binds troponin to move tropomyosin. Sorry, that's an arrow. So that you can form a cross bridge. And once you form a cross bridge, then the myosin pulls the actin in and the muscle contracts. It's kind of a simplified version. If you go back to chapter 10, you have all the detailed steps. The main point here is that you need calcium to form cross bridges, and that you need cross bridges to contract. The more calcium you have, the more cross bridges you can form. Okay, think of playing tug of war, and each cross bridge is a person who's grabbing the rope. Remember, cross bridges are connections between the myosin and actin. Well, the more connections you make, the more force it can pull, right? If you have more people on your tug of war team, you have more hands grabbing that rope, more people pulling, you're going to have more force. So more cross bridges means more force. So you're going to, sympathetic increases the force by increasing the calcium. Now, if you think back to chapter 10, remember calcium is stored in sarcoplasmic reticulum, and it's released from the sarcoplasmic reticulum when you get the nerve signal. So let's talk a little general stuff about muscles here. So in a muscle cell, you store calcium in the sarcoplasmic reticulum, and it is released when you get that signal. Remember the action potential. So it's released when you have an action potential in the T tubule. In cardiac muscle, you have two sources of calcium. So you have calcium that is stored in the sarcoplasmic reticulum, just like you learned about with skeletal muscle. So when that action potential from the node reaches the cardiac muscle cell, it's going to stimulate the sarcoplasmic reticulum to release that stored calcium, just like you learned about back in chapter 10. But you also have a second source. Extracellular calcium will enter the muscle cell. Both of these calciums will be used to form cross bridges. The amount that comes from the sarcoplasmic reticulum is a set amount. You can't alter that. But you can control how much comes in from extracellular. So this is where sympathetic comes in. Sympathetic increases this one. So the more norepinephrine you have equals more calcium that enters, which means more cross bridges, which means more force. And remember, if you have more force, you also increase your stroke volume, the amount of blood that that ventricle is going to pump into the artery. Okay, so we know that sympathetic is going to increase heart rate by letting more calcium in so that you go from -40 to 0 faster, and it's going to increase force by letting more calcium in so you make more cross bridges. Now we need to see how norepinephrine lets more calcium in. Now this part is going to be the same for both the node and the muscle. So again, remember, it's important to keep node and muscle separate. When we're talking about the node, we're talking about heart rate. When we're talking about the muscle, we're talking about force and stroke volume. Don't mix those up. But the way norepinephrine lets more calcium in is the same for the node and the muscle. The result of the more calcium is different. The result of more calcium in the node is you go from -40 to 0 faster. The result of more calcium in the muscle is you get more cross bridges. But we're going to look at the similarity here and how the norepinephrine lets more calcium in. So if we have a cell, we're going to use just a different color here that will represent both the node and the muscle. Here's a cell. Your cells have beta receptors. At least your heart cells do anyway. If you remember from chapter 15, a beta receptor is what norepinephrine binds to. You also have the calcium gates. This is where the calcium comes in. Now remember, if it's a node, the calcium coming in is going to increase heart rate. If it's a muscle cell, the calcium coming in is going to increase force. But they both use the same method to let that extra calcium in. There's another molecule in here called cAMP, cyclic AMP. Think back to chapter 17 when you learned about hormones. Remember, water-soluble hormones bind to a receptor on the surface of the cell. Beta receptor is on the surface. Norepinephrine is a water-soluble hormone. If you've forgotten this, pause the video and flip back to chapter 17 and review water-soluble hormones. When they bind to that receptor on the surface, they activate a second messenger. cAMP is the second messenger. And then, again, flip back to your chapter 17 notes about water-soluble hormones. Second messengers will either activate an enzyme or open a channel or open a gate. So what you have here is norepinephrine binds to the beta receptor. So this is going to be step 1. Norepinephrine binds to your beta receptor. Step 2 is that then activates the C-amp, the second messenger. Then step 3 is the C-amp opens the calcium gate. And then step 4 is extra calcium enters. Then step 5 depends on if it's a node or a muscle. So step 5 in a node would be increased heart rate. Step 5 in a muscle would be the increased force due to the more cross bridges. Okay, so take a minute, make sure this stuff makes sense. Maybe even rewatch this, get this soaked into your head. Next, we're going to look at how some different drugs can act on this. So we have beta blockers, calcium channel blockers, and digoxin. So a beta blocker you may have heard of these, like especially if you already work in a nursing home or a hospital or something where you're taking care of patients and giving them their meds. A lot of older people are on beta blockers. Beta blockers and calcium channel blockers are both drugs that are used to reduce blood pressure. And the way they reduce blood pressure, your blood pressure is related to your heart rate and your stroke volume. So if you can reduce heart rate and stroke volume, you will reduce blood pressure. So they reduce blood pressure by reducing heart rate and stroke volume. Well, you can do that by blocking the extra calcium that norepinephrine causes. Okay, think back to chapter 15. You always have some norepinephrine. You can turn it up, you can turn it down, but you never turn it off. There's always some norepinephrine in your body. That norepinephrine is always causing you to let extra calcium into your heart. If you can stop that extra calcium from coming in, then you're not raising your heart rate, you're not raising your force. So those would therefore go down. Okay, so again, think about that. Norepinephrine is always letting extra calcium into your heart. These drugs work by preventing that. They stop the norepinephrine from letting the extra calcium in. Beta blocker drugs are just like their name says. A beta blocker would come in right here, and it prevents step 1. So if you're on a beta blocker drug, step 1 doesn't happen. Well, if step 1 doesn't happen, neither do steps 3, 4, and 5. So then you're going to reduce your blood pressure by stopping this. A calcium channel blocker works over here. You can still have step 1 and 2, but when it comes to step 3, that's where the calcium channel blocker comes in. It says, oh no, you're not coming in here extra calcium. And it just kind of sits there and blocks it. It doesn't let it in. So again, you don't get the extra calcium in. Okay, then digoxin is our other drug. Up here I have this. If you know old rock and roll, the Eagles and Hotel California, this song reminds me of digoxin. So the calcium can get in, it can receive into the heart cell, but it can't leave. That's what digoxin does. So the point is, you can never leave. That is the part that reminds me of digoxin. So if we look at what digoxin does, digoxin is used to strengthen a weak heart. So like if someone has heart failure and their heart is just weak and it can't pump very well, they're going to have a really low stroke volume. So digoxin is in there to increase the strength, increase the force of the contraction and get that stroke volume up. So the point of digoxin is to increase the force. Well, if you want more force, that means you need more cross bridges. And if you want to have more cross bridges, that means you need more calcium. So remember, you have calcium that comes in from outside. Well, that calcium is constantly moving in and out. This extra calcium that comes in, it then turns around and goes back out between heartbeats. It doesn't just stay in there. So the calcium comes in and then it goes back out. Let's just go ahead and make a fresh picture here. You have calcium comes in and you get some cross bridges. Then after the heart beats, it goes back out. And then it just comes back in the next heartbeat. So it's constantly cycling back and forth in and out. Digoxin comes in right here and it blocks the exit. So that's why it's like the Hotel California. The calcium comes in, but it can't leave. So then with the next heartbeat, more calcium comes in again, right? You're eating calcium as part of your diet, so you're not going to run out. With every heartbeat, more calcium comes in, but it can't leave. So this causes you to build up calcium inside that muscle cell. And all that calcium building up means you're going to get more cross bridges. And by having more cross bridges, you're going to increase the force. So that is how digoxin works to increase the strength or the force of contraction in a heart that is weak. In this section, we're going to compare cardiac muscle to skeletal muscle. So this graph is your cardiac muscle. This one is skeletal muscle. And we're doing this comparison because you already know about skeletal muscle from chapter 10, and we're going to see how cardiac muscle is similar and how it's different. One thing I want to point out here and make clear is that we are talking about the cardiac muscle cells. This is not the nodes. So we're going to be talking about voltages. And we just finished talking about voltages in nodes. But this is not the same thing. This is different. Remember the sarcolemma? So again, if you have forgotten chapter 10, you have to go back and review that. Sarcolemma is the membrane of a muscle cell. And it has action potential and resting membrane potential just like a neuron. So remember in chapter 10 you learned about The acetylcholine, which is the neurotransmitter that stimulates skeletal muscle, is released from the motor neuron onto the muscle. That causes an action potential in the sarcolemma. The action potential goes down the T-tubule. That's what makes the terminal cisternae open their calcium gates, release the calcium, bind the troponin, make cross bridges contract. Okay, so this has voltages, right? You look here, membrane potential, and here's your voltages. This is not the voltages of the node. This is voltages of the sarcolemma. So we're looking at the voltages that happen in a muscle cell. This whole page of notes is going to be the voltages of the muscle cell. Okay, so let's look at what they have in common. They both start with a resting membrane potential of about -90. So here we'll do what they both have in common. Resting membrane potential of -90 is where you start. Then you have sodium comes in, so you have depolarization. And you raise up to +30. So depolarization is the sodium in, and you raise to +30. Okay, then you have potassium goes out, which is repolarization. This should all sound familiar. You learned this in chapter 12 also. Sodium in, potassium out. Here is your repolarization, the potassium out. And that brings you back down to -90. Okay, notice the green area in each of these. We have this green area here, and here we have this little green area. That is the refractory period. So the green area represents the refractory period. Okay, remember in chapter 12 learning about how the action potential moves along the axon and it has to move forward, it can't move backwards. And the reason it can't move backwards is that the sodium gates become locked. Right? Refractory period is the time during which you cannot have another action potential. So you can't start another action potential during the refractory period. Because the sodium gates are locked and they don't open back up until you return to your resting membrane potential. So now's where we'll start to see the difference between skeletal and cardiac. Notice in skeletal that refractory period is very short. You have sodium come in, you go up to +30, then immediately potassium goes out, you're back down to -90, refractory is over. You can see right here, the refractory is ending as soon as you get back to -90. In the heart, you have a very long refractory period. Sodium comes in, you depolarize to +30. Notice there's this plateau. Then you repolarize back down to -90. And then once the voltage, right here, returns to -90, notice the refractory ends. So first we'll look at what causes the longer refractory and then why that is important. So remember with the heart, you're also letting that extracellular calcium in. So remember, that's in the muscle. So go back to your last page of notes. look at the part that says the cardiac muscle cell, the very far right. Remember, extracellular calcium comes in, and that calcium is used to make cross bridges, but it's a positive, it's a cation, so it's also raising the voltage. So in the heart, let's do a color here for when we're talking about just the heart. In the heart only, you have extracellular calcium enters the muscle cell. And we already talked about this extracellular calcium forms cross bridges. And this is what gives you more cross bridges. The more of it you let in, the more cross bridges you get. It also raises voltage because it is a cation that's entering. So in the heart, you have this plateau phase. So right here is the plateau. Notice in the skeletal muscle, the voltage goes up and it immediately drops back down. In the heart, the voltage goes up, then it slowly drops back down. During that plateau, you have calcium coming in and raising your voltage at the same time as you have potassium going out and lowering voltage. So you have one thing raising voltage and another thing lowering voltage at the same time. So they counteract each other. It's like pouring hot water and cold water into a glass at the same time. Well, you're going to end up with warm water. So during this plateau phase, the voltage tends to steady out. You're going to end up with a steady, kind of slowly decreasing voltage. So you have this fairly long period of time between depolarization and repolarization. So in the skeletal muscle, the refractory period is short because sodium comes in, then immediately potassium goes out. You depolarize, you immediately repolarize, you return to -90, refractory is over. In the cardiac muscle, you have sodium come in, you depolarize to +30, then you have your potassium going out, but you also have calcium coming in at the same time. So here we have calcium in, we have potassium out at the same time, so they cancel each other out. And you get this plateau. You don't immediately drop back down to -90. Then when you get to the repolarization, the calcium gates close and you just have potassium going out. And then you drop back down to your -90 and refractory is over. So in the heart has a long refractory. Because during plateau, you have calcium in at the same time as potassium out, and they cancel each other out. Then refractory ends when the calcium gates close. So then the calcium gates close and you are left with just potassium going out, and that is when you repolarize. And when you repolarize, refractory ends. Now let's look at the significance of this. Notice we have this red line here that shows muscle contraction. So here is the muscle contracting, right? And then here is the muscle relaxing. Notice in the skeletal muscle, the muscle contraction and relaxation both occur after refractory is over. Let's get a color for skeletal muscle only. In skeletal only, contraction and relaxation are after refractory. Okay, think of a strobe light. It flashes off and on really fast. That's how you stimulate your skeletal muscles. When you say you lift your arm up and you keep your arm lifted for a whole minute, that's not one nerve signal lasting a whole minute. Nerve signals are a fraction of a second. It's like a strobe light going really fast. You're re-stimulating that muscle over and over again very fast. Once the refractory ends, you can have another action potential. Notice here in the skeletal muscle, you can stimulate it again before it has relaxed. So you can re-stimulate before it relaxes. That is how you keep your arm up for a whole minute. You keep re-stimulating it before it has a chance to relax, and so it stays contracted. And this state of staying contracted is called tetany. Not to be confused with the disease tetanus. Remember, we learned about that in chapter 14. That's a bacterial infection. We're not talking about an infection. Tetany is just a state of keeping your muscle contracted. You do this all the time. Now look at the heart. Here is relaxation. Notice the muscle is already relaxing within the refractory period. Down here is where you can stimulate it again. And by the time you can stimulate it again, it's already relaxed. So that long refractory period, because of the plateau, means that you can't re-stimulate that muscle before it relaxes. So, can you think you can have tetany in the heart? No, you can't. This makes tetany impossible. So tetany does not occur in the heart. You can't re-stimulate it to contract until it has relaxed. It has to relax between contractions. So if we come down here and write this out, skeletal only, the muscle contracts and relaxes after refractory. This means you can re-stimulate before it relaxes. So this allows you to achieve tetany, which just is a sustained contraction. And think about like the planking challenges. Like they say you're in good shape if you can plank for 2 minutes. You're keeping your muscles contracted for 2 minutes in a plank. So you're doing tetany. You're re-stimulating those muscles before they have a chance to relax. In your heart, you have that plateau, stretches out the refractory. So because the plateau is stretching out the refractory, the muscle has time to relax before it can be restimulated. That means you cannot sustain a contraction. It must relax between contractions. This is important because it's during that relaxation that the heart fills with blood. Remember, the heart is a pump, so it has to fill with blood, then contract and pump the blood out. It fills with blood while it's relaxed. So you need this plateau in order to give you a long refractory so that you are forced to allow the heart to relax and fill with blood in order to contract and pump the blood. If you didn't have that long refractory and you could have tetany in the heart, it would seize up. It would contract and then just stay contracted, and you wouldn't actually be pumping blood. You'd just be sitting there frozen in this contraction and not refilling and pumping the blood. So the plateau gives you a long refractory, which means that you are forced to relax between contractions, which means you have a chance to refill. And that's the significance of having the calcium coming in addition to making extra cross bridges. The calcium also provides the plateau. So this extracellular calcium that comes into your cardiac muscle does two things. It allows you to make more cross bridges for more force. And it provides the plateau, which then provides the long refractory, which is what ensures that your heart relaxes to refill.